Welcome. In the last session, I was talking about different types of neurons, and I also told you that the axon is covered by squam cells, and these squam cells produce a substance called myelin. Not all, in all cases, the squam cells will produce myelin. Even though the squam cells are present, they may not produce myelin. And basing on the presence or absence of myelin, so the nerve fibers can be classified into two categories. One, it is called myelinated nerve fibers, myelinated nerve fibers, and another category, unmyelinated nerve fibers. So in these unmyelinated nerve fibers, the squam cell doesn't form myelin. So it does not form myelin. Whereas in case of myelinated nerve fibers, the squam cell forms myelin. So it forms a myelin. Now, where do you find these myelinated nerve fibers? So these myelinated nerve fibers are found in spinal cord and cranial nerves. Okay, I'm sorry, spinal nerves and cranial nerves. So spinal nerves and cranial nerves will be myelinated nerve fibers. Whereas coming to unmyelinated nerve fibers, so these unmyelinated nerve fibers are seen in case of somatic neural system, and it is also seen in autonomic neural system. So somatic neural system or autonomic neural system. So in these you will find unmyelinated nerve fibers, whereas the spinal nerves and cranial nerves will be myelinated nerve fibers, and the transmission of impulse is faster in myelinated nerve fibers. So here impulse transmission is very fast. So coming to the unmyelinated nerve fibers, in this case the impulse transmission is slow. So this is about the two types of nerve fibers, the myelinated and another one unmyelinated nerve fibers. Now let us see the conduction of nerve impulse, that is generation and conduction of nerve impulse. See here, the neurons are excitable cells. So why they are able to show this property is because their membranes are polarized. Their membranes are polarized. So let's see how they are polarized. So we will see by taking the axolemma, axon. So here this is the axon, right? So this is how it is. The axon cell membrane is called axolemma, and the cytoplasm present inside is called axoplasm. So this is the plasma membrane. It is the plasma membrane of axon, and the axoplasm is the cytoplasm of axon, right? And on this axolemma we find protein channels, so that allow the movement of ions to move into the axoplasm or out of it. So we can see here, and there will be different types of ion channels present on this axolemma. So you can see here, there will be sodium channels, there will be potassium channels, and there will be even sodium potassium pumps. So here, coming to these channels, so through these channels the movement takes place passively. The movement takes place passively, that is without using energy, and for this movement it requires concentration gradient. Means on one side of the membrane there should be more number of those ions, and another side there must be less number. So now the ions will move from higher to lower concentrated area. So this is how the channels will function. They will allow the movement of a particular ion. So particular channels will allow particular movement of ions, and they require for that moment they require concentration gradient. And coming to another one called pumps. So these pumps, okay, will help in transporting the materials, or these say suppose in this case ions, from a lower concentration to a higher concentrated area. So that is these pumps actually, right, do the movement of molecules, right, against concentration gradient, against concentration gradient. So that is they move ions against concentration gradient. So that is the function of these pumps. So channels allow the passive movement of the molecules, and for that it requires a concentration gradient. So in this case the movement of molecules will be high concentration to low concentration area, whereas the pumps will move the molecules from low concentration to high concentration. So in this case the example is sodium potassium pump. So in this case it is sodium potassium pump, right, whereas coming to the channels we have is sodium channels, and there are, okay, potassium channels. And these channels are called voltage gated channels, So coming to what actually is this gated ion channel. So the meaning of gated ion channel is it is an ion channel that will open. So it's an ion channel that opens or closes in response to a stimuli. So ion channel that open or closes in response to a stimuli. So here the stimuli is voltage. So the differences in the voltage, right, makes them to close or open and allow the movement of these ions across the axolemma, right? Whereas I already told you that these pumps will move the ions against a concentration gradient. So now on this axolemma we have our say suppose these are, okay, sodium channels. So these are sodium channels, right? So this is a sodium channel and this is the potassium channel, and that is voltage gated. So you may find, okay, gates that I will show you later in detail. Then we may also have that is we also have a third type of channels. So here I am just mentioning, and these are called the sodium potassium pumps, called sodium potassium pumps. So here, right, so the on the axolemma we have sodium potassium pumps. We have right sodium, okay, voltage gated channel. So that's what I'll mention, sodium voltage gated ion channel is present, and another one is potassium voltage gated ion channels. Now in addition to these channels there will be another type of channels which we call them leakage channels. So there are leakage channels. So these leakage channels through which the movement of ions take place freely uh, with respect to the concentration gradient. So here these leakage channels will allow the movement of molecules from high concentration to low concentration. So that is what, so like for example the potassium leakage channels, right? Now out of all these pumps, that is especially the sodium and potassium pump is the one that plays very important role, okay, to maintain a particular concentration on either side and make it a polarized membrane. So here that's what we have something called resting membrane potential. So this resting membrane potential is actually maintained by a sodium potassium pump. I will discuss that one. So right, these sodium and potassium gradients are maintained by are maintained by sodium potassium pump is maintained by sodium potassium pump. So here that tells us the how sodium potassium gradients are maintained on either side of the axolemma. So here that's I'll just mention here the sodium and the potassium, that's what they play an important role. So they play an important role in the formation of resting potential, in the formation of resting potential. So now with this we have understood that the axolemma of the axon has different channels. Okay, so we have come across with the sodium okay, gated channel. So this is sodium voltage gated channel is present. And so this sodium voltage gated channel, so you may see, okay, I'm drawing little bigger. So we have, okay, so these are the gates which help in opening and closing of this channel and allow the movement of sodium ions from outside to inside or inside to outside. And similar way we have another one. Okay, so this is a potassium voltage gated channel, gated channel. And we also have another type, so that is the sodium potassium pumps. So this is a sodium potassium pump. Okay, this sodium potassium pump will okay, move these potassium ions or sodium ions against their concentration gradients. And we also have seen that there are, okay, the potassium leakage channels. So the question comes here, where are all these present? So these are all you will see only in the node of Ranvier regions. So we know that the rest of it is actually covered by, so say suppose in case of the myelinated neuron, so we have here is the squam cell, is the squam cell, is the squam cell, and we find these gated channels in this location. So you will find these gated channels, okay, in the node of Ranvier, node of Ranvier. And the electric impulse will actually transmit not through this squam cell. So this squam cell which forms this myelin, if it is the case, then if it is a myelinated nerve fiber, if it is a myelinated nerve fiber, then the electric charge cannot pass, so this electric current cannot pass through this squam cell. So for that, here the transmission has to take place from one node of Ranvier to another node of Ranvier in this way. So I already mentioned that this is what we call a saltatory movement of nerve impulse. And this we will see in case of right myelinated nerve fibers, whereas in unmyelinated nerve fibers the movement will be straight, and because they don't have this myelin, so I told you already that myelin acts as a insulator, so it acts as insulator, not allowing the nerve impulse to pass across it. So hence it jumps from one node of Ranvier to another node of Ranvier. That what we call it the saltatory movement. So now we have understood that on the axolemma, so presence of these different channels and pumps, okay, will help in maintaining the resting potential and also conduction of nerve impulse. And I told you that the sodium potassium gradients are maintained especially by this sodium potassium pump, which will pump, okay, those sodium and potassium ions against the concentration gradients. So I'll just even mention what actually is this against the concentration gradient. So here let's take the membrane. On this we have is the sodium potassium pump. So this the pumps and all the channels are made up of special proteins that are embedded. Sodium potassium pumps are present, right? Now, let us say that when the neuron is not conducting any impulse, so neuron is not conducting when neuron is not conducting any impulse, then this is what we call a resting potential. So it is resting. So at resting condition, the potential what it has is resting potential. So when neuron is not conducting any impulse, so we call it, okay, in a resting state. Now let us see how is this axon at resting condition. So here this is, I'll take inside. So this inside is axoplasm, and here we have is axolemma. And inside the axoplasm, what are different ions present inside? So I mean when this is at a resting condition. So here there are more number of potassium ions. So inside the axoplasm, I'll just mention here, there is high concentration of potassium ions. Okay, plus along with this, there are negatively charged proteins. So there are negatively charged proteins. Plus there is, okay, low concentration of sodium ions. Low concentration of sodium ions. So uh, that's what here I'll mention. So there are lot many potassium ions, okay, and along with this we have sodium ions. So you can see in case the sodium ions are less when compared with the potassium ions. And here uh, the axolemma is impermeable to the negatively charged proteins. So it is the membrane, that is axolemma, is impermeable to negatively charged proteins. Is impermeable to negatively charged proteins, right? And it is permeable to, means more permeable to these potassium ions, more permeable to potassium ions, and nearly impermeable to sodium ions. Impermeable to sodium ions. So that is the membrane is more permeable to potassium ions, and it's almost impermeable to sodium ions. And these are the conditions, okay, that actually maintain the resting potential. Now what about in the case of ECF? So I told you that outside this axon, ECF is present, and in the ECF, what it all contains. In ECF, it is opposite to that in the axoplasm. So in this case, it has low concentration of potassium ions. So this one has low concentration of potassium ions, and there is high concentration of sodium ions. So there is a high concentration of sodium ions. And now okay, so we have here, I told you that this membrane, that is axolemma, is polarized. It is polarized. Okay, and depolarization actually results in the transmission of impulse. So here in the resting state it is in polarized condition. Now this is maintained by sodium potassium pumps. So here, okay, so these are sodium, okay, potassium pumps. Just mention it. So you can see here these are sodium potassium pumps. Now what these sodium potassium pumps they do is they will be transporting three sodiums outward. So you can see here, I told already, the pump is the one that will, okay, going to pump those ions from lower concentration to higher concentration. So here this sodium potassium pump is going to transport these sodium ions, okay, from lower to higher concentration. Okay, so in the ECF there is low concentration of potassium ions and there is a high concentration of sodium ions, whereas in the axoplasm there is low concentration, okay, of sodium ions. Low concentration of sodium ions is present. So these sodium ions are being transported from, okay, low concentration to high concentration. So here it will pump, okay, three sodium ions to outside. It pumps three sodium ions to outside, right? And right, in response to this it's going to transport inside from outside two potassium ions. So here three sodium ions it's going to pump outside. So let's mention, so three sodium ions to outside, and it's going to pump, okay, two potassium ions into the axoplasm. And because of this, the inside, that is the axoplasm, so inside the axon, it will have a net negative charge on the membrane. So the inside of the membrane will have a negative charge. So the moment of three sodium ions to outside, moment of three sodium ions to outside and two potassiums to inside makes inside the overall net charge to be negative, while the outside it will be positive. So the outside it is positive, the outside it is positive here. Okay, so you can see now this is what I call it a polarized membrane. So inside there is negative charge and outside there is positive charge, and so this okay, polarization is maintained by the sodium potassium pump, right? So by pumping these three sodium ions to outside and two potassium ions to inside. Now these negatively charged proteins, they never ever come out of this, okay, axoplasm, because I said that this is impermeable to negatively charged proteins. So the negatively charged proteins, they permanently remain inside the axoplasm, right? And this movement by the sodium potassium pump will help in maintaining overall net negative charge, that is overall negative charge inside the axoplasm and the positive charge on outside of the membrane. Now the axolemma is said to be polarized. So any difference, okay, again in the movement of these sodium ions will result in depolarization of the membrane. So that will be an initiation and what we call it action potential. So now we are seeing at